Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Central University's Common Entrance Test question paper for the subject Plant Biotechnology conducted in the year 30th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Plant Biotechnology, you can go to my channel Omni Bioentrance, select playlist. In the playlist, search for CUET. In that you will find previous year question papers of plant biotechnology being answered with explanations. So coming to the first question, nodulation in the root hair cell involves signaling cascade. So what is the sequence of the signaling cascade? That is the question. So first will be production of node factors by rhizobium C. Then comes calcium spiking. Then comes activation of cytokinin histidine kinase, then expression of genes and cell division, and nodule formation. Answer is option 2, CDBEA. Then assertion reason question. Soil bacteria such as Streptomyces species produce bulk of antibiotics that help in cell signaling or communication among the members of the same species. Okay, that's correct. Node factor signal produced by root nodulating bacteria are essential for biofilm formation. That is also correct. But Streptomyces is not involved in nodule formation. So reason doesn't explain A. So both A and R are true, but R is not the correct explanation of A. Answer is option 2. Then next one, tDNA integrates into plant DNA and directs cell to multiply producing a tumor. Okay, that's correct. TI plasmid of Agrobacterium tumefaciens include 21 virulence genes. Yes, Vir genes, they help the bacteria to infect the plant cell and transfer its tDNA into the plant genome. So reason is correct, assertion is also correct, and reason explains assertion. So the answer here is both A and R are true, and R is the correct explanation of A. Answer is option 1. Then next one, plant pathogens are known to trigger hormone signaling in plants to show defense response. Reason, jasmonic acid and salicylic acid in plants mediate activation of immune system. That is also correct. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Then next one, the phenomenon where either spontaneously or due to specific treatment the chromosome number is reduced to half in somatic tissues is called, that is called as somatic reduction. Answer is option 2. What is somatic embryogenesis? That is in plant tissue culture where the somatic cells, they differentiate and give rise to embryos. What is chromosome deletion? It is a segment of the chromosome is lost or deleted. What is chromatic restitution? That is the process where the chromosomes that have already been fragmented or disrupted due to various factors, they are restored to their original structure. That is called as chromosome restitution. Then next one, spontaneous variations that arise in haploid plants with gametic chromosome number are called as. So they have specifically mentioned gametic chromosome number. So it must be gametoclonal variation. Clonal variation, it is seen in identical individuals, that is genetically identical individuals you see clonal variations. Mutagenic variation is that occur due to exposure to mutagens, and somoclonal variation is seen in plants regenerated through somatic cells in tissue culture. So the answer here is gametoclonal variations. Then next one, the pH of the culture medium is usually adjusted between So it is adjusted between pH 5 to 6. Answer is option 2. Then next one. The steam sterilization in an autoclave is achieved at. So it is achieved at 121 degrees Celsius for 15 to 20 minutes. Answer is option 4. Then which of the following are micronutrients used in culture medium? Nitrogen, phosphorus, all these are macronutrients. They have asked specifically micronutrients. So molybdenum, zinc and copper. Answer is option 3. Then next one. Which of the following is not a limitation of ovary culture? They have asked not a limitation. It leads to the production of haploid plants. That is why you do normally ovary culture. So it is an advantage of ovary culture. So it can lead to the production of haploid plants. So first statement is not a limitation. Answer is option 1. Then it is successful only in a small number of species. That's correct. That's a limitation of ovary culture. Higher levels of plant growth regulators may suppress gynogenesis. That's also correct. The frequency of responding ovaries and the number of ovaries is generally quite low. That is also correct. So the answer here is option 1. Then they have given two statements. Statement 1. The optimum stage of pollen for anther culture varies with the species. Of course. For some species, the optimum stage is just before or after the first pollen mitosis. Yes. While for others, it may be the early binucleate stage. That's also correct. Pollen culture is independent of the culture conditions and can be performed at any temperature and photo period. That is not correct. It is highly dependent on specific culture conditions including temperature. So statement 2 is not correct, but statement 1 is correct. So the answer here is option 3. Then next one. A HUP plus gene, that is uptake hydrogenase gene, is used which helps in recycling hydrogen produced during nitrogen fixation. That is correct. Then it also reduces the energy lost as hydrogen. Then it increases ATP availability and it also enhances nitrogenase activity, increasing nitrogen fixation. So that are the functions of HUP plus gene. Then statement 2. Rhizobic strains with HUP plus gene cannot fix nitrogen more efficiently. Now, HUP plus gene, it enhances nitrogenase activity, increasing nitrogen fixation. So it can fix nitrogen more efficiently. So statement 2 is false. So answer is option 3. That is statement 1 is true but statement 2 is false. Then next one. Match the following function of drug and the drug they have given. Viral fusion inhibitor, that is phoscarnet A4. Blocks viral penetration, that is amantadine B1. Inhibits neuraminidase, that is oseltamivir C2. Protease inhibitor, it is ritonavir D3. So answer here is option 2, that is A4, B1, C2 and D3. Then next one. Tissue culture raised plants are subjected to specific cultural regimes to make them capable of being transferred to soil is called. Okay, that is called as acclimatization. Somatic embryogenesis is production of embryos from somatic cells in tissue culture. Regeneration is, that is you regenerate the plant in tissue culture. Pre-treatment is pre-treatment of the explant with surface stirlens. All these are pre-treatment. So the answer here is acclimatization. Answer is option 1. Then assertion, reason, questions. The reduction of atmospheric nitrogen to ammonia depends on the production of a protein called as leg hemoglobin. That is correct. The transfer of ammonia from the rhizobium to the host plant involves a complex cycling of amino acids. That is also correct, but it doesn't explain the assertion. So A and R are true, but R is not the correct explanation of A. Answer is option 2. Then next one. Capsid protein gene 6 from tomato yellow leaf curl virus provides enhanced resistance in susceptible tomato hybrids. Of course, that's correct. Transformed plants recovered more effectively from disease than the untransformed plants. So when these capsid protein gene 6 from tomato yellow leaf curl virus was used for plant transformation, it was found that the transformed plants recovered more effectively from the disease than the untransformed plants. Why? Because capsid protein 6 provide enhanced resistance in susceptible tomato hybrids. So reason explains the assertion. So both A and R are true, and R is the correct explanation of A. Then next one. Generalized step for surface sterilization of explant material are. So first you have to wash the explant thoroughly under tap water. Okay, E. Then comes rinse with 70% ethyl alcohol. Then treat with 0.1% HgCl2. Then wash with double distilled water. Then you place the explant on culture medium. So answer here is E, B, A, C, D. Now if you know that the last step of this whole surface sterilization is culturing or placing the explant onto the culture medium, then you can happily go for the third option, because in third option only D is coming in the end. So the answer here is option 3. Then next one. Sequential steps involved in the production of haploid plant through anther culture. Okay, so first you have to desiccate the anther, squeeze out the microspores. So E will be the first one. Desiccated anthers are pressed to squeeze out microspores in a sterile beaker containing liquid medium. That will be the first one. Then the suspension with anthers and microspores are filtered through a nylon sieve. Okay. Then centrifuge the suspension to collect microspore pellet. Microspore are your pollen grains. Then microspores are inoculated on a solid or liquid medium maintained at 25 degrees Celsius for an 16 to 8 hour photo period. Then embryoids are formed and produce haploid plantlets. So here the sequence will be E, A, B, D, C. Now here also, as I have mentioned previously, if you see that embryoids are formed and produce haploid plantlets. Here the question is the production of haploid plants. So C should come in the end of the sequence. So only in 3 C is coming in the end. So that must be your answer. But you have to read the other statements also. Even if you find that C is coming in the end and that is the answer, you have to still read the statements. Then antibiotics and their result. So vancomycin, it prevents transpeptidation, A3. Then tetracyclines, they block, or I can say they prevent protein synthesis inhibition, B4. Quinolones block DNA replication and polymyxin B cell membrane disruption. So the answer here is option 2, A3. They have forgot to mention B over here. Here it should be B, B4. C1 and D2. That was a printing mistake. Then next one. Protoplast fusion can be effectively used for the production of CMS line and has the advantages. Only one step is required. That's correct. CMS is cytoplasmic male sterile lines. That is correct. Nuclear genotype of the cultivar remains unaffected. That's also correct. 100% of the progenies of somatic hybrids will be CMS. That's also correct. Mitochondrial DNA of the progeny resemble the mitochondria of the parent. Because it cannot be, because protoplast fusion, the mitochondrial DNA of the progeny can be either recombinant, heteroplasmonic, or maternal or paternal origin. So recombinant means it is a combination of parental mitochondrial DNAs. Heteroplasmonic, that is also somewhat similar to the recombinant, but it is different. It is a mix of parental mitochondrial DNAs. Then maternal or paternal, it is means that it is either of the maternal origin or of the paternal origin. So statement D is not correct. Then protoplasts are treated with iodoacetamide to activate mitochondria. No. Iodoacetamide, it is used to inhibit mitochondrial DNA replication. It will eliminate mitochondrial DNA and then create CMS lines. So statement E is also not correct. So the answer here is option 2, ABC only. Then next one. The major benefits of micropropagation include rapid multiplication and maintenance of uniformity. Of course, A is correct. Multiplication of disease-free plants. That's also correct. Production of chimeric plants. No, that is not a benefit of micropropagation. It is an expensive and labor intensive. It is not expensive and labor intensive. So A and B only. Answer is option 1. Then next one. Which of the following pre-treatments do not promote androgenesis? So androgenesis, it is the process where haploid plants are produced from male gametes. So irradiation with X-ray and gamma rays, yes, you can get haploids. Cold treatment also can give rise to promote androgenesis. Then glutamine and sugar starvation, yes, also. Temperature of more than 60 degrees, that is not used to promote androgenesis. So 4 is the answer. Then next one. The shoot multiplication medium is designed to promote axillary branching such a medium usually contains. So they usually contain high cytokinin to auxin ratio. This is because high cytokinin auxin ratio, it will promote axillary bud break and it will enhance shoot multiplication from axillary buds, and thereby it will increase the branching and it will also reduce apical dominance. So first one is the answer, high cytokinin to auxin ratio. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of plant biotechnology, you can go to my channel Lumini Bio Entrance, see the playlist. From the playlist select CUET. In that you will find previous year question papers of plant biotechnology being answered. Thank you.